Okay, so that is on whole genome sequencing. And then I will go into the metagenomics. So metagenomics means, looking into microbial communities. So I think 2022 and 2023 was a breakout year for hi-fi metagenomics. There has been, multiple publications that, uses our long-read sequencing to look at both 16S as well as shotgun metagenomics using hi-fi sequencing. So the number of papers that you can see, after like 2022 and 2023 and 2024, the number of hi-fi metagenomics publications have been increasing by a lot. I think one reason is, of course, researchers are actually see the value of our long-read sequencing for metagenomics, as well as, of course, the declining cost of our sequencing uh, uh, out, output itself. So that's why, more and more people are actually, jumping into using hi-fi sequencing for metagenomics work. So the first metagenomic technique that I want to talk about is, of course, 16S sequencing. So we know that, for 16S, all bacteria actually have a 16S gene, right? And usually we try to identify the variation in this conserved genes so that we can actually we can actually identify the species or genus of this particular bacteria itself. So targeted sequencing of this 16S is actually a much cost-effective way to actually assess the composition of the metagenomics communities. So I think usually for short-read sequencing, a lot of people are actually still looking at just V3 and V4 region, sequencing this particular region. But for hi-fi sequencing, we actually advocate to actually do a full-length 16S sequencing. So, because by doing full-length 16S sequencing, you are able to actually detect more nucleotide differences, for better taxonomy resolution. And we can actually distinguish, down to species and strain level compared to, just looking at V3 and V4. Yeah. So, just to show you a publication from George Weinstock, one of the leader of the Human Microbiome Project, right? They actually showed that, looking at different regions of the 16S gene itself is biased towards certain bacteria. And for example, for V4 itself, if you look at this particular image here, right? If So the redder you go, which means the more unclassified percentage, unclassified microbes that you have, unclassified bacteria that you have in your sample itself. So you can see that for V4, there are many, it's so red, which means, many of the bacteria are actually unclassified. Whereas if you are doing, a full length, for example, V1 to V9, you can see that most of the bacteria you can actually classify them into the clade itself. Yeah. And you can see that there is also another paper that actually shows that, will actually comparing between PacBio full-length 16S sequencing compared to V4 as well as synthetic long-read full-length 16S. Means, you use short read, but you combine them, you sequence through, and combine them to actually form a synthetic 16S. So uh, it shows that there's 99.7% of the sequences can be assigned to species level when we are using PacBio hi-fi sequencing compared to LookSeq, which is the synthetic long read full length 16S compared, and also V4 itself. And there's also another publications that have shown that there's a high correlation between full length 16S compared to shotgun metagenomics. So in this particular paper itself, they showed that uh, they found 140 species classified in both data sets, and 88% of them assigned to 16S and 74% of them assigned to the shotgun reads. So and even at genus level, the correlation actually increased to 90%. So by doing full-length 16S sequencing, you have data that is similar to what you can get from, doing shotgun metagenomic profiling already. And some of the interesting applications, of course, we know that people are doing gut microbiome as well as human gut microbiome, and we do have customers that are using our full-length 16S to do um, animal biome as well for dogs. So they do have, kits that they've developed for dogs as well as for cats to actually look at their gut microbiome health itself. So I think this company is in Australia, and we do have this paper. So if you are interested, we can actually share to you. So for 16S itself, right, we do have two different techniques that you can use. One is on the standard monomer protocol, whereas the other one is the Kinex protocol that I mentioned earlier, which actually, make our uh, cost of 16S to go as low as the short read sequencing. So for standard monomer means, we are actually sequencing just one 16S gene in one single um, SMRT well in the smart cell itself. So this is suitable if you're not running a lot of samples. But if let's say you are running more than 192 samples on the Sequel 2E or 384 samples on the Revio, we will recommend you to use the Kinex workflow. So what the Kinect workflow does is it will actually combine. So we know the 16S the full length of the 16S sequence is around 1.6 kb, right? But as I mentioned, our hi-fi sequencing generates reads that are up to 15 to 25 kb. So if for every well we are just sequencing 1.6 kb, we are wasting a lot of the potential that you can actually sequence, right? So what we do in this Kinect workflow is we actually concatenate or we combine multiple 16S fragments together to form one single, long read itself. And then this one single long read, we will actually, uh, we will prepare this library, and then we will actually subject it to sequencing. And then after that, we will use bioinformatics pipeline to, demultiplex them and, to generate each segmented reads itself. So by having this Kinex 16x kit, we are able to actually, increase the throughput by 12-fold. And by doing that as well, you are able to actually, save a lot of cost in terms of you can multiplex a lot of samples on one smart cell itself. So that's why we are able to actually generate, the low cost, which is, that is, comparable with what you are running on short read right now.